Sensory information is thought to be encoded by assemblies of activated neurons in the brain. Different sets of neurons are stimulated by various sensory stimuli, forming distinct ensemble representations, also called patterns of activity. Yet, it remains unclear how the ensemble representation changes over time in various behavioral contexts. In addition, it is also unknown whether populations of neurons receiving similar inputs but having different projection targets exhibit different forms of ensemble plasticity. In my laboratory, we are studying the olfactory bulb as a model system. Others activate sensory neurons in the nose, leading to an excitation of output neurons of the olfactory bulb called mitral and tufted cells. Others are known to activate specific ensembles of mitral and tufted cells. In our recent work, we demonstrated that manipulating representation of output neurons of the olfactory bulb can alter learning in naive mice. Yet, it is not known if these representations are plastic and how they would evolve in time during different behavioral contexts. In addition, it is not clear if mitral and tufted cells, two populations of output neurons that target different cortical structure, would behave similarly and whether the representation would be similarly plastic.
In order to address these questions, we performed chronic two-photon calcium imaging in awake behaving mice. We expressed a genetically encoded calcium indicator, specifically in mitral and tufted cells, to monitor their odor responses over time. Since mitral and tufted cells are located in different layers of the olfactory bulb, we can image them separately by changing the focal planes. This is an example field of view with many neurons changing their fluorescence intensity in response to odor stimulation. A cell can display either increase or decrease in calcium, or excitation or inhibition, respectively. The response timing can be either during or after the end of odor stimulation. Collectively, both cell types show similar response profiles with relatively balanced excitation and inhibition. We then asked how sensory responses change over time under different behavioral contexts. Mice were either passively exposed to the same odorants for consecutive days, or trained to perform an odor discrimination task under the microscope. After several days of training, mice reliably learned to discriminate a pair of very similar odorants. When the rewarded odor, or S+ is applied, an expert mouse responds with licking to get water reward. In contrast, when the non-rewarded odor, or S- is applied, it suppresses licking. Do ensemble representations change differently between passive experience and active learning conditions, and between mitral and tufted cells? Here we schematically present ensemble representations as images of flowers. In naive mice, the ensemble representations of S+ and S- are quite similar, reflecting the high similarity of the two odorants applied. In tufted cells after active learning, and in mitral cells after passive experience, both representations were reorganized, but the similarity between S+ and S- remained relatively high. In mitral cells after active learning, the representations of both stimuli were again reorganized, but strikingly, the similarity between S+ and S- significantly dropped, leading to improved pattern separation. In summary, we found two forms of ensemble plasticity. One is improvement of pattern separation, which is specific to mitral cell and active learning, and the other is reorganization of sensory representations, which is independent of cell types and behavioral contexts. At the moment, we know little about the circuit mechanisms underlying these different forms of plasticity. Our computational model, however, suggests that the cortical feedback to the olfactory bulb may play an important role. This would be an interesting starting point for the future research.
In olfaction, it has been a hot topic of debate whether mitral and tufted cells are doing the division of labor. We believe that our results provide the first direct evidence that mitral and tufted cells display remarkable differences during active learning, but not during passive experience. The olfactory system might be maximizing the capacity of information processing by having two output channels with distinct properties. Our results show the existence of different forms of ensemble plasticity that varies with behavioral context and output neuron population considered. These observations demonstrate the ability of brain circuits to reorganize and to adapt to various ethological conditions.